Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 12th November 2020. And as all of you know, we are into the revision mode and we are revising all important chapter number 8, cell structure and function. And cell, as we defined, is the unit of life, basic unit of life. And we said cell is a membrane bound structural and functional unit of life that contains a cytoplasm and cell organelles. And basically Robert Hooke was the first to observe cells in a cork, but what he saw was actually the cell walls enclosing spaces left by dead protoplasts. Anthony van Leeuwenhoek first saw and described live cells. Robert Brown later discovered the nucleus. And of course, cell theory was jointly put forward by Sleeden and Swan in 1839. In its modern form, it includes the following three principles. What are those? All organisms are composed of one or more cells. Cells are the smallest living things, the basic units of organization of all organisms. Cells arise only by division of a previously existing cell. On the basis of nuclear organization, cells are classified into two types: prokaryotic cell and eukaryotic cell. As I said yesterday, pro means primitive, karyon means nucleus. Cell that has got a primitive nucleus, not very clear nucleus, we say nucleoid. On the other hand, eukaryotic, eu means very clear, karyon means nucleus. A cell that has got very clear, well-defined nucleus is eukaryotic cell. And basically, the cells that are present in plants are called plant cells, and the cells that are present in animal cell or animals are called animal cells. So in animal cells compared to the plant, cell wall is absent. Cell is composed of plasma membrane or cell membrane, and it is composed of lipids that are arranged in bilayer. So bilipid layer. The lipid component is mainly composed of phosphoglycerides. Later it was found that protein is also present in cell membrane. Ratio of protein and lipids varies in different cells. So basically an animal cell membrane is made up of lipoproteinaceous layer, lipids as well as proteins. And in animal cells, chloroplasts are absent. And the animal cell often possesses many small vacuoles, and centrioles are found in animal cells, and in case of plant cells, centrioles are usually absent except in lower plants. Golgi apparatus is either localized or consists of well connected single complex. So basically the cell contains a cell envelope, and we must be thankful to two scientists, Singer and Nicholson, in 1972 proposed fluid mosaic model. This is very famous. According to this model, the quasi-fluid nature of liquid enables the lateral movement of protein within the bilayer of lipids. So as you are able to see here, you can see the bilipid layer, lipid bilayer. Okay, two layers are there, and in between the proteins are sandwiched. So you got this cholesterol part of it, then sugar part of it, all that. So basically the plasma membrane or plasma lemma forms the outermost boundary of cytoplasm, which separates it from the extracellular environment and controls the entrance and exit of molecules and ions, and thus helps to maintain the difference in ion concentration of the cytoplasm and the surroundings. Several types of models have been put forward to explain the structure of biomembrane. The most accepted model is fluid mosaic model. And this fluid mosaic model of plasma membrane was proposed by Singer and Nicholson. According to this model, biological membranes are semi-fluid or quasi-fluid, we said. This is quasi-fluid or semi-fluid structures in which lipids are and intrinsic proteins are arranged in mosaic manner. That means you got two layers of fats, and in between the two fatty layers there is a layer of proteins, and these protein molecules are intrinsic within, they are arranged within. And how do they arrange? They are arranged in the form of a mosaic flooring. According to this model, lipid molecules in the membrane are arranged in the form of bimolecular layer, so that their hydrophilic polar heads, hydrous means water, philic means love for the water. So hydrophilic polar heads constitute the outer and inner surface, while hydrophobic tails, water again, phobia, fear of water. Hydrophobic tails are buried in the membrane, as you are able to see here. The proteins are arranged in two forms. One is the extrinsic, that is peripheral proteins, that is spectrin, and intrinsic or integral proteins, as you are seeing intrinsic proteins. And these integral proteins are sandwiched inside the two bilayer lipid layers. The integral proteins which run throughout the lipid bilayer are known as tunnel proteins. Example, these we can say glycoproteins. So this is how the fluid mosaic model is made up of. The main function of plasma membrane is the transport of molecules across it. So main function, transportation, making the molecules move from one area to another area. In that, two forms are there: active transport and passive transport. The transport that involves an expenditure of energy by the cells. Active means ATPs are spent. Passive, the cells do not spend ATP in passive transport. Active transport, it occurs against the concentration gradient, and that is why ATP molecules are to be spent. Whereas in passive, the transport is always along the concentration gradient, so there is no need to spend extra energy. It's a fast, rapid process, active one. It's comparatively, passive one is slow process. So the movement of water from higher concentration to lower concentration by diffusion is called osmosis. The cell wall is present in plant cells and fungi. Algae have cell wall made up of cellulose. The algal cell wall is made up of cellulose. Further, the minerals like calcium carbonate also will be present. In other plants, it consists of cellulose, hemicellulose, pectin, very tough one, and proteins. So primary cell wall of young plant is capable of growth, which diminishes in mature cells. Secondary cell wall is formed on inner side of the cells. Plasmodesmata connects the cytoplasm of neighboring cells. These are plasmodesmata is finger-like structures that connect the neighboring cells. Endomembrane system of cell includes ER, endoplasmic reticulum, Golgi body or Golgi complex, lysosomes and vacuoles. These are all part of the endomembrane system. And as you are seeing this, you can see the endoplasmic reticulum. Since ribosomes are present on the endoplasmic reticulum, this is called RER, rough endoplasmic reticulum. If ribosomes are not there, then we say SER, smooth endoplasmic reticulum. And you can see the plasma membrane is getting connected to nuclear membrane, and that forms the endoplasmic reticulum. So you got a nucleus, nuclear pore, ribosomes, and of course the one that is not having ribosomes is smooth endoplasmic reticulum. So basically the endomembrane system grouping of some membrane organelles which function in close coordination with one another, that is endoplasmic reticulum. They divide intracellular space into compartments as you are seeing here. Basically it makes two, luminal and extraluminal. Luminal is inside endoplasmic reticulum and extraluminal is cytoplasm. These can extend from nuclear envelope to plasma membrane as you are seeing here. And the RER provides surface for protein synthesis, because rough endoplasmic reticulum is providing space for ribosomes, and ribosomes are responsible for protein synthesis. The endoplasmic reticulum is composed of the following three kinds of structures. One is the cisternae, which occur in those cells which have synthetic roles, tubules, which are involved in lipid and sterol synthesis, and vesicles, that are membrane bound vacuolar structures. The ER is of two types, as I said just now, smooth endoplasmic reticulum and rough endoplasmic reticulum. So you got SER and RER. And this rough endoplasmic reticulum consists of cisternae. It has ribosomes attached on the cytoplasmic surface. RER provides surface for protein synthesis. The proteins in endoplasmic reticulum lumen are processed and are packed in membrane bound vesicles for storage or export or for further processing in Golgi apparatus. Formation of nuclear envelope and plasma membrane. Smooth endoplasmic reticulum is involved in fat and steroidal hormone synthesis, the function of SCR. And it is also involved in detoxification of drugs and xenobiotics using cytochrome P450. And that's what the importance of endoplasmic reticulum. So basically it's scattered in the cytoplasm. RER bears ribosomes on its surface. RER is involved in protein synthesis and secretion. SCR does not bear ribosomes on its surface. SCR is involved in lipid synthesis. This you must remember, lipid synthesis and steroidal hormones. And then comes the Golgi apparatus. It was first observed by Camillo Golgi. That's why the name is Golgi apparatus, near nucleus. They consist of many flat disc-shaped sacs or cisternae. So cisternae are disc-shaped sacs. These are stacked parallel to each other. Golgi apparatus performs the function of packaging of materials and its transportation. A number of proteins synthesized by ribosomes are modified in cisternae of Golgi apparatus. Golgi apparatus is the site for synthesis of glycoproteins and glycolipids. This is important. Function of Golgi apparatus. So the Golgi complex is in the form of a parallelly arranged interconnecting system of cluster of smooth membranes having central stack of flattened sacs or cisternae. It has two distinct phases: forming phase, that is cis region, and maturing phase, trans region. Process, package, transport, release of secretory proteins is the function of Golgi body. A glycosidation of lipids and glycosylation of proteins to form glycolipids and glycoproteins. Golgi apparatus helps in the recycling of broken plasma membrane during endocytosis. That means it is mainly not only for synthesis of glycoproteins and glycolipids, it's also for cleaning purpose, excretory in nature. And then we go to the next one, that's lysosomes. Lysosomes are membrane bound vascular structures formed by the process of packaging in the Golgi apparatus. They are rich in hydrolytic enzymes. This is important. Lipase, protease, carbohydrates, active at acidic pH. These enzymes are capable of digesting. Lipase means lipids. Lipids get dissolved by lipase. Protease, proteins get digested. Carbohydrates get digested. And of course nucleic acids. So these are the functions of lysosomes. They were first discovered by Christian D. Doo in 1955, Lysosomes store the hydrolyzing enzymes of the cell. They are also called acid hydrolases because these digestive enzymes usually function in acidic medium or pH 4.5. Lysosomes arise from bud of vesicles of the Golgi complex at the trans phase. So, in injured and dead cells, the lysosome membrane ruptures spontaneously, releasing the enzymes that dissolve the weakened cells. Therefore, the lysosomes may be called as suicidal bags of the cells. This is important for UPSC NDA as well as NEET exam. So, lysosomes are called suicidal bags of the cell. Why is it so? Because they just break themselves off and release the enzymes. They are particularly abundant in certain cells like white blood corpuscles, macrophages, Kupes cells, and in secretory cells. So they are basically autophagic vacuoles. Hence, lysosomes play also a crucial role. We will continue further in our next class. Until then,